Today's lecture is the third lecture in the series dealing with immune dysfunction. As I mentioned with the previous lectures, we spent the entire semester learning how the immune system functions, and with the last three lectures, we've been focusing on what happens when parts or components of the immune system does not work properly. The first in the series dealt specifically with hypersensitivity, which is also referred to as an allergy. This is an exaggerated immune response that has deleterious effects and can cause damage to the individual. The hypersensitivity response is a problem with recognition in that components or antigens are being recognized that are not truly a potential threat, and an immune response is being elicited. The second lecture focused on autoimmunity, and autoimmunity is an inappropriate response of the immune system against self-components. Here too we have a recognition problem. Self-antigens are recognized as a potential threat, and as such an immune response is elicited. Autoimmune disorders dominate when tolerance mechanisms fail. So in that lecture, we also spent time talking about tolerance and how tolerance mechanisms exist to protect an individual from potentially self-reactive lymphocytes. So again, when tolerance mechanisms fail is when we see traditionally autoimmune disorders. Today's lecture is going to focus on immunodeficiencies, and this is really just a situation where there is a deficiency in the immune response. Simply some aspect of the immune system or response does not work. And that is divided into two categories, which we will focus on today. Primary immunodeficiency, which is a condition that results from a genetic or a developmental defect in the immune system. And secondary immunodeficiency, which is an acquired condition resulting in a defect in the immune system. There are hundreds of documented chronic primary immunodeficiency disorders that have in fact been documented. If you go to this link, if you're interested, you can see just a subset of some of those primary immunodeficiencies, and on this slide you see a few examples. We'll come back to these examples and look at them in a little more detail, just so you have an understanding of what's happening in each of these cases. Because there are in fact so many different well-documented situations or chronic disorders of primary immunodeficiency, oftentimes what scientists do when there's lots and lots of information is try to categorize them. We've seen this as a reoccurring theme throughout the semester. And so with primary immunodeficiencies, we have the same situation. They are oftentimes categorized by the type of problem associated with the immunodeficiency. In this particular slide, you have a highlight of a number of different immunodeficiency disorders, and you can see that they are sort of color-coded by boxes. In the orangish color, orangish red color, you can see those that are considered phagocytic deficiencies. So any deficiency where the phagocytic system is not functioning properly fits into this particular category as a phagocytic deficiency. In the green boxes, you can see some of the disorders that are labeled or categorized as humoral responses. These are all situations where there is a problem with antibody function or antibody production. On the far right, you see one box labeled DeGeorge syndrome, which is a reddish, pinkish color. This is a cell-mediated category. Now, there's not only one disorder that falls into the cell-mediated category. However, this slide is just a summary overall. So those disorders that have problems with cell-mediated functions are going to be those that have problems with T-cell responses. And then lastly, what you see here in the purple or bluish colored box are those that are referred to as combined categories. This is where one or more cell type is affected by the immunodeficiency. As I mentioned a slide ago, there are over 400 documented primary immunodeficiency disorders. And what you see here is a very short list of some examples. But what I like to do is go through some of these examples so that you have some understanding of what is happening in the deficiency and why it's considered a deficiency overall. So the first one is A-gamma globulinemia. This is a B-cell defect. Individuals that suffer from this particular disorder typically produce very low levels of IgG and oftentimes have an absence of all other Igs. Obviously, this is again a B-cell defect in that antibody production is not really occurring in these individuals. This particular disorder has been determined to be due to a mutation in a gene that is known to code for Bruton's tyrosine kinase. This particular protein, when it is produced and functions properly, plays a role in signal transduction in B-cell maturation and development. So individuals with a mutation in this particular gene that codes for this particular enzyme have problems with B-cell maturation and B-cell development, hence the absence of Igs and in some cases very low levels of IgG. The next immunodeficiency on the list is known as DiGeorge syndrome. This syndrome, in fact, is due to a deletion of a portion of chromosome 22. So these individuals have other problems than just immunodeficiency. They typically have heart defects, thyroid problems, modified facial appearance, heavy eyelids, oversized ears. So again, there's a lot of additional components associated with this particular syndrome because of the deletion of a portion of a chromosome. However, in this particular case, the deletion of the portion of the chromosome plays a role in not allowing for proper thymus development. And in some situations, there's a complete absence of the thymus in these individuals. As you can imagine, someone who has a problem with thymus development or a complete absence of the thymus is going to have very severe problems associated with T-cell development. So oftentimes these individuals have a depression, meaning very low levels of T-cells or no T-cell responses at all. And again, you can imagine that that's going to be very severe in an individual that's afflicted with this particular disorder. The next one is Chidiac-Higashi syndrome. This happens to be a problem associated with phagocytes. There is a protein known as the LYST protein, LYST, which is normally involved in the regulation of intracellular trafficking. You might wonder why a problem with intracellular trafficking would be considered an immunodeficiency. Well, if you think about the phagocytic process, we know how phagocytosis occurs. We talked about it in depth and in detail, step by step. How do all of these components move within the cell in order for fusion of different components to take place? It's all due to intracellular trafficking. If there is a mutation in the gene that codes for this protein, it turns out that targeting of proteins to the lysosome does not occur properly. So in this particular case, again, this is a phagocytic defect in that phagocytosis does not occur properly. There have been, in fact, approximately 30 different mutations in this gene identified for this particular disorder. The next one, known as LAD, or leukocyte adhesion deficiency, is going to be a genetic disorder where there is a mutation in a gene for a protein known as LAD1. LAD1 is part of the integrin system and is essential for leukocyte adhesion. We know that leukocyte adhesion is extremely important for inflammation, specifically in getting target cells to the target location during an inflammatory response. Individuals that have this particular disorder have a very large decrease in the recruitment of cells to the site of inflammation because of the problem with the adhesion protein overall. The next disorder I want to focus on is Wiskott-Aldrich syndrome. Wiskott-Aldrich syndrome is fits into the combined category of immunodeficiency, and that's because both B cell and T cells are affected in this particular syndrome. There is a protein known as CD43, which is a cytoskeletal glycoprotein in lymphoid cells, and it is required for the assembly of actin fibers. Essentially, this particular protein is required for signal transduction. In individuals with this disorder, there's a mutation in a gene that codes for this protein, and hence signal transduction in both B cells and T cells is dramatically reduced or inhibited overall. The last one on the list is known as Severe Combined Immunodeficiency Syndrome, or SCID. This is the most severe form of primary immunodeficiency that has been documented. It actually is a family of disorders that are all involved in lymphoid development. In individuals with this particular disorder, they typically have very low number of lymphocytes, often have failure of T cell mediated responses, as well as B cell responses. These individuals typically don't have thymus development that occurs properly. And in these individuals, it is a very severe problem in that recurrent infections typically occur, and this is usually fatal in these individuals. If any of you have seen the movie or heard of the story, The Boy in the Bubble, this particular story focuses on a young man who had this disorder overall. Now, it turns out because it is a family of disorders, there have been mutations identified in several different genes that can contribute to the disorder. I'm just going to highlight one or two of them. One of them is a gene that codes for an interleukin-2 receptor. It turns out that if this particular receptor for interleukin-2 is not produced properly, typical cytokine responses are not received. Hence, the development of both B cells and T cells does not occur properly. Another example is a mutation in a gene that codes for a protein known as JAK3. This is a protein that is an enzyme in the signal transduction cascade. So here too we have problems with B cell and T cell development because signal transduction is inhibited. By far the most common or well-known secondary immunodeficiency due to an infectious disease is the disease known as AIDS, acquired immunodeficiency syndrome. This is due to the human immunodeficiency virus. The disorder was first reported in the early 1980s, where individuals in large metropolitan areas were coming into the hospital with very unusual types of infections, things that doctors were not necessarily normally seeing or treating. Oftentimes these were opportunistic fungal pathogens, again, things that doctors were not normally seeing and treating in individuals. And so this raised a big question as to what was going on in these individuals overall. With this particular disorder, there is in fact a decrease of cellular immune responses, specifically helper T cell responses. And that's because this virus targets the helper T cells and infects specifically helper T cells. And so there is a decrease and a depression in helper T cell responses. We know that this particular disorder has reached epidemic proportions, and we know that the exact infection mechanism is still not completely understood. However, there is plenty of research going on with this disorder. We do know, however, that transmission of the disease, the virus, occurs via passing of bodily fluid. Things like unprotected sex, infected blood via IV drug use, and can also be passed on from mother to child. So let's look at the human immunodeficiency virus in a little more detail. It turns out that this particular virus falls into the category of viruses known as retroviruses. Retroviruses have their genetic material composed of RNA. So you can see that in this particular virus, there is a single-stranded RNA genome. There's actually 2 copies of the genome found packaged in every virus. In retroviruses, the RNA must be reverse transcribed to DNA by an enzyme that is virally produced known as reverse transcriptase. And we'll talk a little bit more about that in just a minute. In addition, we know a little bit more about the virus itself and the structure of the virus. We know that there are two proteins known as P17 and P24. Both of these are proteins that form what's known as the protein coat. There are two protein coats in this particular virus, and those are highlighted on the slide, shaded in the green color and in the yellow color. We also know that this particular virus is considered what's known to be an enveloped virus, meaning that some viruses are able to cover themselves in host phospholipid bilayer, and that is exactly what you see in this particular situation. Interestingly, because this virus is an enveloped virus able to coat itself in host phospholipid bilayer, there are also components of the host phospholipid bilayer that are found on the exterior surface of the virus, such as the MHC protein. This makes the virus very easily missed by our immune system. It evades the immune system because these host components are found associated with host plasma membrane components. And again, it makes the virus very hard to be detected by the immune system. In addition, we know that the outer component contains glycoproteins 120 and glycoprotein 140. Glycoprotein 140 allows for the fusion of the virus with the host membrane that it is infecting, and glycoprotein 120 allows for the interaction with the receptor on the host cell as well. As I mentioned in the previous slide, the target cell of infection for this particular virus are helper T cells. Specifically, the glycoprotein 120 protein binds to the CD4 receptor that's found on helper T cell surfaces. And the last thing that I want to talk a little bit about with this particular virus is that every viral component has packaged in it multiple proteins that are important for the virus to function. In this case, I've highlighted 3 of them. There is a protein known as integrase. This is a protein that is essential for integrating the viral genome into the host genome. There is a protease that is important for processing of viral proteins. And reverse transcriptase, like I mentioned at the beginning of this slide, is an enzyme that's required to copy the RNA-based genome into DNA so that integration can occur into the host as well. This virus has been studied rather extensively, and so we know a lot about the genomic information in this particular virus. What you see here is the viral genome that has been mapped out as far as the proteins that are coded by the genes in the viral genome. I won't ever ask you to know all of these, but we do in fact know all of the genes and what they code for in this particular virus. There are genes that code for nucleocapsid proteins. These are proteins that are found on the surface of the virus. Envelope glycoproteins, again, these are portions that allow for the protein coat to be formed. Enzymes, like we discussed in the previous slide, that are packaged in with every single virus. Regulatory proteins, some auxiliary proteins as well. So let's take a little closer look at what's happening with infection of a target cell. The target cell, again, is the helper T cell, specifically because the viral glycoprotein 120 binds to CD4 on the surface of the target cell. And you can see that in step 1. In step 2, we have the glycoprotein 41, which mediates fusion of the virus with the membrane of the host cell. In step 3, the nucleocapsid enters the cell. This nucleocapsid contains the viral genome and the enzymes that were packaged into the virus. In step 4, what happens next is the release of the viral genome and the enzymes that were packaged inside. The next step, step 5, is where the viral reverse transcriptase copies the RNA genome and synthesizes a DNA component. At this point, there is an RNA-DNA hybrid molecule that has been produced. Step 6 involves RNA degradation and then second strand synthesis so that there is a complete double-stranded DNA copy of the HIV genome. Step 7 then allows for integration of the double-stranded DNA copy of the HIV genome to occur into the host genetic information. And this occurs with the help of the enzyme that is virally produced, known as integrase. Now, the double-stranded DNA viral copy of the genome that has been integrated into the host can lay dormant inside the host genome for a long period of time. What allows for this dormancy and the length of the dormancy is still not 100% understood. We do know, though, however, during the infection process, the next stages are going to be to activate that provirus and produce more viral components. In this stage, transcription factors allow for the transcription of the provirus to occur, both the single-stranded RNA genome component as well as messenger RNA components. Step 2, the viral RNA is exported into the cytoplasm. Step 3, divided into A and B. A is what's happening specifically with the messenger RNA. They are associating with ribosomes to allow for the synthesis of viral proteins. And then step B, you can see those viral proteases. Again, the protease that was originally packaged in with the virus cleaves and modifies these proteins so that they can be packaged into new viral components. Step 4, you have viral particle assembly, where all of the pieces and the parts of the virus that have now been produced in the host can be packaged. And then in step 5A, you have viral envelope formation taking place. And then lastly, 5B, release of the new virus that has been produced. Lastly, a virus such as this that has a very good evasion mechanism for evading the host immune system is a very difficult target for scientists and doctors to treat for individuals who are afflicted with this particular disorder. The best targets are going to be targets that are viral specific. So things that you see highlighted in the boxes are all viral specific components and are in fact targets of current drug therapies for treating the disorder known as AIDS and the virus known as HIV. So, for example, inhibiting the reverse transcriptase would be a fabulous target because if we could inhibit the reverse transcriptase, the DNA copy of the viral genome would not be produced. Inhibition of integrase would also be a good target because this specifically would inhibit the integration of the double-stranded DNA, if in fact it were synthesized, from getting integrated into the host genome. Target drugs could inhibit fusion. This one's a little bit tricky because of the receptor that it's binding to. However, this is a good target as well. And then lastly, inhibition of the protease makes a lot of sense because it is the protease that then takes the precursor proteins, cleaves them, and prepares them for assembly into new viral particles. So again, ideally, what scientists would like to target are viral specific components so that host cellular components are not necessarily affected.